this is where we were. So today we're going to talk about antibodies, and I'm going to go back to this slide. This is in the chapter 4 PowerPoint. So like if you want to, it's number 62 in the chapter 4. So if you just want to pull that one up as kind of a reminder, you know, you don't want me to have to draw all that. So I know we're in chapter 5, but if you want to have that figure, it's in the chapter 4 PowerPoint number 62. So we'll go back and forth a little bit. All right, so a request was made for 2 jokes. So I will do 2 jokes today. So today's joke is, what's the difference between a tuna fish and a guitar? You can't tune a fish. What do you call a cow with 2 legs? Lean ground beef. Okay. I didn't say the second one was going to be any good. I kind of like it though. You need a bad joke? I thought. No, dad joke. Oh, dad, is that a dad joke? What makes it a dad joke?
The really cheesy quality.
Mine are super cheesy, but I don't think they qualify as dad jokes. I don't know. All right. So a couple of people were asking, do not forget that our exam is coming up, right? So we have an exam. Today is the 15th. We're right here. We're Friday. We're supposed to be starting chapter 6 today, which we will. And so not next Monday, but the Monday following will be exam 2. Exam 2 will have chapters 4, chapter 5, and then however much of chapter 6 we cover up until that point will be on the exam. Excuse me. So we tend to be a little bit behind most semesters, but you know, I think that's okay this semester. I think it's all right. So we'll just have an exam on whatever we've covered from chapters 4 to 6 up to that date. Okay. All right. Questions about that? Good. All right. Don't forget to do the seating chart, please. Everybody do the seating chart. And I'm going to switch back. Okay. So we have been talking about protein purification methods, right? And so one particular type of protein that we use in biochemistry so much are called antibodies, right? And we talked about antibodies in chapter 4. So if you want to refresh your memory, and I'll come back to this, the diagram for the structure of an antibody is on slide 62 of chapter 4. So antibodies, if you've taken anatomy and physiology, some people yes, no, not yet. Some people have. Okay, are produced by B cells. Where do we find B cells? Ready? The spleen. The spleen is where the B cells are made. Sorry, y'all. Okay, let's see. Bone marrow. Well, so lots of cells actually do get made in the bone marrow, and then they travel to the spleen where they mature. So if you're going to find the mature B cells, you're going to find them in the spleen. Yes, they are blood cells, and so they originate bone marrow, but they develop in the spleen. So what's really cool about your immune system is that every single B cell makes a particular type of antibody, right? So if you want antibodies to coronavirus, you want antibodies to the flu, antibodies to whatever it is, each individual B cell only makes one kind of antibody. So do you only have one type of B cell in your body? No, you have a ton. So what's the job of any given antibody? To bind to foreign things, right? So B cells go through a selection process Because what happens if they recognize self? Like if my B cells recognize my proteins, my antigens, what do I now have? An autoimmune disorder, and that's really bad. So they go through a typical screening process so that doesn't happen. So B cells can make a wide variety of antibodies, and it's all about the light chains and heavy chains. So if you go back here real quick, what are the heavy chains? Whoa. The heavy chains. What are they in this diagram? They're blue, right? So this is one heavy chain. Do you see how an antibody has 1, 2 heavy chains? How many light chains does it have? 1, 2. So it has 2 light chains, and they're in red, and it has 2 heavy chains. Okay, so where on this antibody do we recognize the antigen? At the antigen binding site. So we have that in both the heavy chain and the light chain. So you see where it's kind of like zigzaggy? That's your antigen binding site. So you have half of the binding site that's in the heavy chain and half of the binding site that's in the light chain. Yeah? So why are these zigzaggy and these straight? The zigzag represents what? The variable region. The straight part represents what? Constant. Okay. So for given antibodies, the constant domain remains pretty constant in sequence. The variable domain is very variable, changes a lot. Why does the variable domain have to change a lot? Yeah, because it has to have unique sequences so that it will bind to unique antigens. Otherwise, it's only going to bind to one thing and it's not going to do you any good. So you have to have a variable region. Okay. So there are 2 classes of light chains. Okay. Anybody know what these are? The first one is lambda, LAMBDA. Okay. And then kappa, right? Okay, so how about these? So you have 5 classes of heavy chains. Heavy chains, we have mu, alpha, delta, epsilon, E, Pi, SLON, right? SI. Okay, well, you whatever, y'all get it. And then gamma. Have you ever heard that when people talk about antibodies? No, right? But what have you heard? Have you heard of IgM, IgA, IgD, IgE, and IgG? That's what it's referring to. Lots of people hear those terms but have no idea what it's referring to. It's referring to the heavy chain. What's the constant region of the heavy chain? And there are 5 different ones. So this is mu, this is alpha, this one's delta, this one's epsilon, this one's gamma. Okay. So that's kind of something you never know you really needed to know. Immunoglobulin. Immunoglobulin, and then it names the heavy chain. Okay, so we know that our immunoglobulins have multiple domains, right? We have the variable domain, which if we go back, right? If we go back here, our variable domain can be variable heavy and it can be variable light. So that's why when we list it, we say it's variable light and variable heavy. What those things stand for. Then you have the constant region. So you have a constant light. So let's look at this real quick. We have a constant light domain right here. There's only one. How many are there in the heavy? 1, 2, 3. So if we go back, there's one light chain constant region, and then there are 1, 2, 3 heavy chain constant regions. See why it's important to look at the structure so you understand what it means? So then we divide the immunoglobulin into different fragments. So remember we said that you can treat these antibodies with a protease. And what does the protease do? It cuts right here. So what do you end up with? A small fragment and another small fragment, right? And so you actually end up with 2 of these top fragments, and we call these 2 the Fab fragments, right? And we call this the FC fragment. Okay. So our Fab fragment contains the antigen binding site. So that's always the section that's really, really important. So now, we said antibodies bind to antigens. An antigen can be really big, right? So there are actually multiple sites on one antigen that antibodies can bind to, and we call those sites epitopes. So within, let's say, the coronavirus spike protein, the spike protein itself is the antigen, but the spike protein's huge. So you can have multiple antibodies binding to the one spike protein because there are multiple epitopes on the spike protein. That makes sense? Yeah. So this is an example, right, of like, let's just say this was a spike protein. Well, there's 4 different epitopes, and at each of those different epitopes, different antibodies can bind. But what's the whole point of an antibody? Why are antibodies awesome? They bind to foreign things, so what?
Block those antigens from binding
to their targets. So like if you have a coronavirus molecule, right, and it uses those spike proteins to bind to the ACE2 receptors on airway epithelial cells. So if you cover the spike proteins with antibodies, you hide it, right? It's like those spike proteins don't exist, even though the virus is inside of you. And so then it can't bind to the receptor, it can't get into the cell, it can't initiate an infection. But you would have to have a ton of antibodies to make that happen, right? So sometimes you don't have enough to inhibit complete infection. So what's the other thing that they do? That's true and very, very important. What's the second thing? They're like little flags for the immune system. Like, hello, other immune response cells. This thing is foreign. You need to come and eat it and degrade it and get rid of it, right? So it's kind of twofold what they do for you. One is hide those foreign particles from being able to do anything, and then also to tag it and to say, hey, this is bad, get rid of it, right? It's a what? It's a bouncer. It is. Absolutely. Kick out those foreign particles. Okay. So there are 2 different kinds of antibodies. Have y'all heard of this before? Monoclonal and polyclonal antibodies. Anybody? No? All right, all new. Awesome. So what does poly mean in general? Many. What does mono mean? 1. Okay. So what does clonal mean? Yeah, basically copies. So if you have something that is, let's start with monoclonal. If you have an antibody that is monoclonal, remember how I said every B cell only makes 1 antibody? So that means it's a homogeneous collection of antibodies. They're all made by 1 B cell. So they only recognize 1 epitope on 1 particular antigen, right? So it's made by 1 B cell, right? Polyclonal, on the other hand, is a mixture of lots of different antibodies. They are going to recognize one antigen, but because that antigen has multiple epitopes, right? You can have multiple antibodies that recognize that epitope, that recognize that antigen, right? So this is made from many B cells. Okay, so this is one antigen, right? Antigen. This is also one antigen, but this is one epitope. This is many epitopes. Yeah. And there are all kinds of reasons why we use antibodies in science. And we're going to talk about a lot of them today. Not all of them, but a lot of them. So it's really important to understand how the different antibodies are made. Now, I am not talking about making antibodies for like treating coronavirus. And I'll talk about, after I explain this, I'll talk about why that has to be different. But this is making antibodies that we're going to use in a science experiment on the bench. Okay, we're not going to put it into a person. We're doing an experiment on the bench. So what we need an animal that will make antibodies. It could be a rabbit. It could be a mouse. It could be a donkey. It could be a chicken. There are all kinds of different animals that you can use, but you immunize this organism. And you do it a couple of times over about a 6-week period. What do you immunize them with? What does that mean when I say immunize? I'm injecting them with an antigen. So you inject organism with antigen. Where did I get that antigen? Not made by a B cell. Remember, antigen is on something foreign. It's all right. No, it's lots of new terms. Lots of new terms. Is an antigen relatively large or small? It's just small. It's relatively small. So there are two ways that I can make it. So I make the antigen via one of two ways. I can do a synthesis. What do we call that? I can actually synthesize a protein. Do you remember we talked about that last time? Solid phase synthesis. Right? You're making a short peptide. The other thing that I can do, we talked about a couple of chapters ago, but I can clone in my polypeptide sequence, my peptide sequence into an expression system. So remember we talked about a plasmid and you put in your sequence of interest and you could have a bacteria express. We use the example of insulin, but does it have to be insulin? No, you can put anything you want in there. So I'm going to put this into a bacteria, clone in peptide sequence into bacteria to express the peptide, which is AKA the antigen. Right? Anywhere where you see peptide in here, really, that's the antigen. Because all the antigen is a little protein sequence. And on that little protein sequence, different antibodies can bind. Okay? So yeah. No, like the antigen would be like, for example, the coronavirus spike protein, right? So it wouldn't be from the animal. It would be something foreign. Oh, no, not really. It's just like getting a vaccine, right? That's what you're doing. You inject into your body a little portion. It's not a whole virus, right? It's just a little piece, or it's an attenuated virus that's expressing this little spike protein or whatever it is, right? We do it all the time. We get vaccinated all the time. So it's not. So it's basically mimicking a vaccine. And what's the whole point of a vaccine? Generate antibodies, right? Generate an immune response to whatever it is you were injected with. Well, guess what I want this rabbit to do? Make antibodies to my antigen, right? It's probably not going to be an antigen the rabbit ever actually sees in normal life, but it's for my science experiment. So then what I do is I collect a blood sample from this rabbit. Inside the serum of the blood, we have a great big mix of antibodies, right? Because one antigen has multiple epitopes. So that rabbit's B cells are going to make all kind of different antibodies. And so I collect the serum, I collect them all. And I purify them with affinity chromatography, right? I can have beads that I know will interact with a target. What's my target? If this is affinity chromatography and my antibodies should target my antigen, what should be attached to the resin in this column chromatography? My antigen, exactly the same thing that I immunized with. We immunized the rabbit, immunized with the antigen, right? And then after I collect the blood, here in my column, I have a bead, not a bed, a bead that's attached to the antigen. So then which antibodies should I catch? Only the antibodies that are to my antigen, right? You got to think about this as an organism. How many different antibodies are floating around in this organism's bloodstream? So many. I don't want them all. I only want my antigens that are specific to I only want my antibodies that are specific to my antigen, right? So that's why I want to wash it over a column that has my antigen attached to it. And then you can elute it with varying salt concentrations and things like that will make it release. So basically the non-specific antibodies should wash right through. They shouldn't bind to the column. And then only when I add some kind of eluting solution, like high salt concentration or something like that, will then my antigen specific antibodies be released. Now I have this pool of antibodies that are specific to my antigen. Now, are they all the same? No. So this is really a mixture of antibodies, right? So they all bind the antigen, but at different what? Epitopes. And I heard somebody else say levels. And that's true as well, because it can bind at the same place, but some can have higher affinity than others. So you can have that as well with different epitopes and affinities. Yes.
Now with the antibodies you get from the would they be bound to antigens?
No, not anymore, because here they were bound here. At this point, they're bound. But then I'm going to add some kind of a solution that's going to release them, right? So usually it's a salt concentration thing. And they can't bind in high salt concentrations and they elute out. But yeah. Okay, so now I have these purified polyclonal antibodies. All right, that's polyclonal. Now, how do we make monoclonal? and then we'll talk about why we're going to use each of them in particular. So monoclonal, we start in a very similar fashion, right? And here we're just showing a different organism. This is a mouse. We do the same injection of our antigen. So we're injecting our antigen, same as before. Right? So that's no different. But instead of harvesting whole blood, what we harvest are B cells. Where do we get those B cells? From the spleen. Right? Okay. Now, we are going to fuse these antibodies, right? In each antibody, they colored them differently to show that they're making different antibodies. We are going to fuse individual isolated B cells with immortalized tumor cells. B cells, if we put them in culture, they will die. They'll only last for a limited amount of time. But tell me about a tumor cell. They will keep going and keep going and keep going and keep going and keep going. Like HeLa cells, right? Anybody read The Immortal Life of Henrietta Lacks? But you heard about it. Yeah. So they collected those cells in I want to say like 40 something. Way. I have used them in the lab. They have been around for a really long time because they're immortalized tumor cells. So by fusing these isolated B cells with an immortalized tumor cell, we create a new cell. And we call this a hybridoma, right? It's a hybrid between the B cell and the tumor cell. So it's going to live forever, but it's going to make antibodies for us. Cool, right? Now, what we do is, and I hate the way that they did this. You're going to have this collection of hybridoma cells. And what you do is you dilute it. You dilute it. So much so that you can put it on a plate that has individual wells, right? And you get one cell in each well, right? So in each cell, you only have one of these hybridoma cells. You understand where I'm going with this? So now I have a place where I can culture each of these individual hybridoma cells because each individual hybridoma is creating a unique antibody. So now I can screen these cultures and I can test them and I can proliferate them so that I can create a never-ending stock of antibodies that recognize one single epitope on my one antigen. Isn't that awesome? And then all I have to do is collect the fluid that the cells are growing in, because what do B cells do? They make antibodies and they send them out into the serum, right? Well, the serum is basically the culture medium that you're growing your cells in. So all you have to do is collect the liquid that your cells are growing in, put them some fresh, and they're good to go again. Pretty awesome. So you have a never-ending supply of monoclonal antibodies. Really cool, right? So why the heck would you ever use polyclonal? Do you know how expensive this is? This is very expensive to do. Very expensive. Very time consuming. So there are drawbacks and there are perks to both, right? If I do a monoclonal, I'm only recognizing one epitope on one antigen, and that's it. That can be really, really powerful in science. Or I can more cheaply and more quickly create a polyclonal. Because if I do create a polyclonal, what happens when my rabbit dies? I got to do it again with another rabbit, right? So you're never going to get exactly that same mix of their polyclonal again. But here I can keep going back to this cell. It's an immortalized cell. It's going to live forever. Good. All right. So now what do we do with all these antibodies? My gosh, we've talked about antibodies for so long. One of the major techniques that we use antibodies in is called Western blotting. Anybody ever done a Western blot? Heard of a Western blot? Yeah. Okay. So I have done an experiment, right? And I want to know in my purification process, right, whatever I've done, where is my protein associating with another target protein? Well, how am I going to know that? They're too small to see, right? Remember I said, what's my job, mixing clear liquids? How do I know my protein's there or not? Well, I can look at size, but that's not insanely specific, right? I want something that's very, very specific. Antibodies can get me that. So I can run a gel that has whatever sample I want on it. And then I can use antibodies to recognize exactly the protein that I want. So let me show you how it works. You need 2 antibodies to make this work. So this we've done before, right? We've run an SDS page gel. We've stained it, and the things that are blue are proteins, right? What's this first column? That's my standard that I know the mass of, right? So that's my molecular mass markers. All these others are my samples. So what I can do is I can actually put a filter membrane underneath my gel and run an electrical current through it. So my proteins that were in the gel will actually migrate and fuse to this membrane. So now I have a filter membrane that has everything on it, but I want to know which of these spots contains my protein of interest. I put an antibody that will detect my protein of interest. This primary, it's called the primary antibody. This primary antibody is typically monoclonal, right? Because I want it to be very, very specific, right? So my primary antibody can be like a mouse and I'm going to say my protein of interest is actin, right? So it's an antibody that was produced in the mouse and will recognize an epitope on actin. Okay, does that make sense? Now, I wash my membrane. I make sure to get rid of anything that's not bound. And then I add a secondary antibody. This secondary antibody is typically polyclonal. So this is my primary mouse. My secondary is going to be a rabbit anti-mouse. Why do I want it to be an anti-mouse? So it binds to the mouse antigen. It doesn't matter what part of it, it doesn't make a difference. As long as it will bind to a mouse antibody, it'll work. So I could have made a donkey anti-mouse. I could have made a chicken anti-mouse, right? It all would have worked as a secondary. Now the secondaries are a little bit special because not only is it a secondary, but it is also fused to an enzyme. And so if you put a substrate into your wash fluid that the enzyme that's attached to the antibody can recognize and metabolize, you can turn that substrate into a fluorescent product. So now, do you see how everything was lit up before? Because that's just proteins. But the only 2 that the antibodies bind to and recognize are 2 that had actin in it, because we see them fluorescing. So it's a really powerful way to look at a mixture of proteins and specifically identify which sample has exactly my protein of interest in it. It's really cool. That's not the only way that you can do this. The other thing that you can do, I thought I had the slide in here, but you can have like live cells and you can fix them and you can kind of like punch holes in the membrane and you can put in these antibodies in the same sequence and you can look at it under a microscope and you can actually see it in fixed cells. And I'll show you a picture of that in a minute. So it's really, really powerful. But sometimes you don't have an antibody to your target. Like, what if you don't know really what it is yet? You don't know what epitope is going to be exposed. You just don't have it. What you can do is you can actually insert your gene of interest with an extra tag in it. So either a flag tag or a MIC tag, there are many different ones, but you put this little tag at the very beginning of the protein or you put this little tag at the very end of the protein. And the tag is really just a sequence of residues. And now a flag tag, you go out on the market, you can buy antibodies to flag tags for really, really cheap. Right? And so basically I've created an epitope on my protein of interest. Now, what's the problem with this? What's that? You have to know which protein you have. So you have to be able to express your protein in this cell type with the tag inserted into it. So you need to know which protein it is to start with. But I am messing with the amino acid sequence of my protein. What happens if my N-terminus is really important for interacting with a protein target in the cell? It could stop working, but maybe it's not really important. That's usually why you put it on the N-terminus. You try one on the N-terminus and one on the C-terminus and see if you get different results. And if you do, you shouldn't use that method. But so it can be really useful, or it can be more hurtful than it is useful. Okay. So we can do antibodies where we look at things in a gel, right? That's everything that we've done here. This is an example of a Western blot. This sequence, like at the very end, when you get this as a picture. But you can also do it in cells. And so you basically fix your cells to a glass slide and you punch holes in them, right? So they can't go anywhere. And then you put fluid on top of them that contains the primary antibody, you wash it, you put the secondary antibody, you wash it, and then you go and view them, right? And so let me show you a picture of what you see. So this is an example of a picture of a cell where they've actually tagged two different proteins, right? So they've done two different primaries, two different secondaries, and two different color fluorescent dyes. So the green here, which you can't see with a black arrow, the green is actually targeting a protein called phalloidin, Illoidin, which is an actin binding protein. And so what's actin? I keep saying it's my favorite protein ever, but that's not a good answer. It's a cytoskeletal structural protein that gives your cell its three-dimensional shape, right? So it's like an internal skeleton for your cells, but it's not the only cytoskeletal protein. The red in this case is another type of cytoskeletal protein. And so what you see here is that different cytoskeletal proteins have different functions within the cell. Can you see clear differences of where the red and the green are? Absolutely, right? I see some overlap, sure, that's the yellow part, but I can see exactly where these proteins are in the cell, which is really useful information. Yeah, it's really cool.
In the structure that you see the fluorescent 458.
Yeah.
What is the part where the binds don't contact on the aromatic
Here,
No, the, like the aromatic ring connected to that, carbon.
this one,
Yeah.
this one. It's a, so this is part of the fluorophore that's going to allow for that fluorescence. And it's going to change depending on if it's bound or not, and whether it's excited or not.
So does that just mean it's a part of it, but it's not connected?
Yes and no. But you don't,
Is it one of those weird things where it's like bound to the ring?
yes. Yes, it interacts with the ring.
Oh,
Like a ferrocene experiment we did in organic lab or something like that?
Yep.
so it's
Yes,
bound
it-
to the ring, but not necessarily a particular atom.
Correct.
Okay.
Correct. Okay. So last assay with, no, there's two. I take it back. There are two more with enzymes. So, so the other thing that's really, really powerful, right? I want to know if someone has a current infection, let's just say, right? So I can take a sample from them. Maybe I'm going to take a sputum sample, right? You got a hockaloogie, right? And I want to know if coronavirus is in there, right? That's a legitimate question. So what I can do is I can take a plate, and each plate has 96 wells. So you can detect coronavirus, you can detect flu virus, you can test for a bunch of different things at once. And in each of the wells, you have a particular antibody that is stuck to the bottom of the well. And we call this the capture antibody. And so you take this sputum sample and you dilute it, and then you put a little bit in each of the individual wells. Where you have an antibody that will recognize, let's say this is the coronavirus spike protein or whatever it is, right? This antibody will catch it, right? And you wash all of them to get rid of anything that's not bound. So in all the wells, some of the antibodies have captured something and some have not. In the ones that have captured an antigen, there is another antibody that binds to the same antigen, but at a different epitope. Okay, so this is called the detection antibody. Now, the detection antibody might be a mouse, right? Remember how we did that? So this one can be a mouse, right? And then I'm going to say, if it's actin, I'm looking for actin. So this detection antibody is another mouse anti-actin, but this is going to be epitope, T-I-T-O-P, epitope 2. This is going to be epitope 1. E-P-I-T-O-P-E, epitope 1, right? So they're recognizing the same thing, but at a different location. So now you have a sandwich, right? You have a detection antibody, you have an antigen, and then you have a capture antibody. The last thing that you do is you add that secondary antibody. So this is just like in a Western blot. This is our secondary antibody. And this is a rabbit anti-mouse. So the mouse part is specific to the detection antibody, right? So now I can see, because this one is covalently linked to an enzyme like horseradish peroxidase, that will change a substrate from being non-fluorescent to being fluorescent. And I can look at each well, and whichever well lights up fluorescently has my antigen present. So I can tell, is this a flu infection? Is this a coronavirus infection? Is this whatever infection? So this is a very fast way to find very low levels of your particular protein of interest. All right, one more. All right. So sometimes you want to know, not only is my protein there, but what is my protein actually interacting with in the cell, right? Because binding partners are insanely super important. Otherwise, nothing works in the cell, right? So how do I figure out, I know my protein's there, but what's it associating with? Who's it hanging out with? And so for that, we do immunoprecipitation, but really it's co-immunoprecipitation, right? Because you want to find out who it's hanging out with. So that's where the co comes from. So what we do is we purify our sample, but we're very careful not to disrupt its interactions with its binding partners. And we use an antibody to recognize actin, our protein of interest, right? And so we're going to pull down this whole complex in that same bead. Remember how we ran it over an affinity column sort of deal? You could either run it over a column, or you can attach the antibody to the bead and just spin it, right? How heavy is a bead compared to a single cellular protein? Bead's really big. Bead's really big. So the beads will come down very quickly, very, very quickly. And you can get rid of the supernatant and just keep the pellet. And then you run that sample on a gel. So now I'm only putting in the things that are associated with actin. And then I can actually cut out individual bands and I can go send them to a mass spec, right? But what do I have to do before? Because a protein's pretty big. I need to cut it up with trypsin or chymotrypsin or something like that. Then I can do my mass spec. And I can, using mass spec, identify the exact amino acid sequence that were in the proteins that associated with actin. And so I can identify, okay, actin's interacting with these proteins. Now, if you have cancer, actin no longer associates with these proteins or whatever it is, right? So that's a really, really powerful tool. But what did you need to make that happen? You needed an antibody. You need an antibody that is specific to your protein of interest. So how powerful are antibodies in biochemistry? Like insanely, insanely important. You can use them for so many different things, right? And this is another example. I feel like we see this all the time, but we don't talk about it. Did we invent antibodies? No. Are we very good at inventing stuff? No, we kind of suck. But what are we really good at? Manipulating what we find in nature. That is what real science does. You know, Mother Nature is a very powerful instrument that can do things that we have no idea. And so if we just start to understand, this is why basic science research is so important. It was basic science research that discovered the CRISPR system. Without that discovery, that wasn't what the two ladies who got the Nobel Prize, that wasn't what the Nobel Prize was given for. The Nobel Prize was not for the discovery of the CRISPR system. The Nobel Prize was for the manipulation of the CRISPR system to change your genome, right? So, I don't know, I just feel like I have to explain those things and I get so excited about it. And this is why we get behind. But it's okay, right? Y'all are okay with it. All right, so this is what we were supposed to start with today. And what time is it? 11:32. All right. It's okay. Whatever we get to in chapter 6 will be on the next exam. I'm not going to cram it down your throats. I don't think that that's the right answer. Okay. So what are we doing in chapter 6? Chapter 6 is protein function. We've talked about how proteins fold. We've talked about how we use proteins in biochemistry, how we purify proteins in biochemistry, but basically how do they work is this one. So this is how proteins work. And there are 5 major classes of proteins. Now, not all proteins are going to fit in one of these 5, and some proteins are going to fit in more than one. But these are like the most common classes of proteins inside of the cell. And then what we're going to do is we're going to take an example from 3 of them and illustrate how important conformational changes are in order for proteins to function, and how the interactions of these proteins with other proteins and with themselves really facilitates their function. So we'll use those 3 as examples. So here are our 5 major functional classes of proteins. Let's kind of talk about each one individually. So proteins that function in metabolism. What kind of proteins do we think these are going to be? Yeah, they're going to be catalysts. They're going to be enzymes, right? Because metabolism is all about changing substrates, from substrates to products. That's what enzymes are all about. And what is metabolism about? Either breaking something down or building something up, right? So we're talking about enzymes being reaction catalysts. C-A-T-A-L-Y, catalyst. That's not right. So to control metabolic flux, right? Should you be breaking things down? Should you be making things? That's metabolic flux. Okay, structure. What do we think on these? Like we're going to maintain the cell shape. We're going to maintain the cell structure, that kind of stuff, right? So we're going to maintain, we'll say cell integrity. Maintain cell integrity of shape, right? You can say shape or structures. S-T-R-U-C-T-U-R-E-S. Sorry, y'all. Didn't get a PhD in spelling. All right, transport. These are super important, right? And they really do work hand in hand with structure very often, right? So in transport, we want to always be facilitating movement, right? So it's T-T-A-T, facilitate movement. Molecules, right? Because we have to move molecules within the cell. If you make something in one compartment and you need it in the other side of the cell, you have to have something that's going to ferry it. Like how do vesicles get from one point to another? Well, something's got to actually move it, which I have a really cool video of that, which I don't know that we'll get to today, but we will. So facilitate movement of molecules. It's going to be within the cell, right? And what else? And the cells themselves, right? We like to move from location to location. So yeah, and outside. We're just going to put outside of cells. Okay, the color. Cell signaling. So the ligands are going to interact with receptors. So receptors are a big part of cell signaling. And do you remember what we called this in the very beginning? Yeah, receptors participating in signal. Right, so these are I like to call these molecular, M-O-L-E-C-U, molecular switches, right? Because they turn functions off and on inside of the cell. Okay, and then genomic caretakers, right? We want to make sure to maintain the integrity of our genetic information, right? So is it being copied properly? Is it being stored properly? Right? All of that is what genomic caretakers do. So maintain integrity. Integrity and accessibility, right? Because if you can't access it in order to express it, you have a problem. Well, and decondensing and condensing, going from heterochromatin and euchromatin, that kind of stuff, right? And accessibility of genomic information, genomic info. Yeah. So now we kind of know all the different things we're going to talk about. So here's just some ideas, some examples of the different classifications of our proteins. So what was number one? Metabolic enzymes. So what do you see in metabolic enzymes, right? You're going to start with some material that comes into the cell, and you're going to have these enzymes that are going to catalyze a change, right? Maybe they become phosphorylated. Maybe we're talking about glucose being metabolized into generate ATP, right? So sometimes you're going to consume ATP, sometimes you're going to produce ATP. What was our second one? Structural. So here's structural. We really have two kind of big groups. You can have intracellular structure. So we've talked about actin, and we've talked about microtubules and things like that in the very beginning. But you also have to think about the structural proteins that are made in the cell and then excreted, right? So we call that the extracellular matrix, or the ECM. Have you heard of that before? Yeah. Okay, so collagen, all those things that are excreted into there. What was number two? Number three. Transport proteins. So here are transport proteins that span the membrane, that allow for things to come into and out of the cell, right? Then we said we had signaling. So here's our signaling, right? Cell signaling proteins. We have something that binds our ligand on the outside, that will then translate that via a signaling mechanism into a change of a cellular function. And then the last one, genomic caretakers. So everything involved in replication, transcription, translation, all that stuff. And that is what we will talk about next time. Look, at least we got 2 slides in, 3 slides in. That'll be okay. Y'all are learning, right? Okay. Think we have Zoom need anything? You're welcome, Tammy. All right, if nobody needs anything else, I'm gonna go ahead and end the meeting.